Hello, my name is Dallin Thewison,
I'm Kent Charrington.
And I'm Logan Waite.
and today we're going to be talking about Lesson 12 and the group problems.
Okay, so for question 1, it pretty much gives us a list of terms here, and then on part A it asks which of these terms describes or is an example of a regulatory protein. So for here, because it's just asking for the actual proteins, we put activator, repressor, and the CAP. For part B, it asks which of these terms describes or is an example of a small effector molecule. We put CAMP, an allolactase, an inhibitor, or an inducer. Okay. For part C, we put it asks which of these is an example of a sequence found within the DNA. We put the +1, the -10, the operator, the lac A, and lac Z. For part D, it asks which of the above is not involved in the lac operon. We found that was the inhibitor. Okay. So for part E, it asks which of the above is an example of a structural gene. As we know from the reading, it's the lac A and the lac Z.
Okay, part 2 is fill in the blanks. The lac operon is regulated by two inducers, cyclic AMP and allolactose. The binding site for sigma factors and RNA polymerase is called LAC-P, while the CAP site is the binding site for the activator that regulates the lac operon. The enzyme adenyl cyclase converts ATP into CAMP, and the -35, -10, and +10 sequences are found within the LAC-P portion of the lac operon.
So for part E, the LAC-L gene encodes the lac repressor, while the lac-Y gene encodes lactose permase. And then attenuation can regulate gene expression in bacteria by causing transcription to terminate prematurely. And then lastly, in part G, it says an E. coli strand that contains 2 copies of the lac operon is called a merozygote strand.
Okay, so then on question 3, it gives us just a bunch of information about, the cells that we're dealing with. But essentially, the IPTG is our representative of an allolactose, okay? And as we know from the readings, the allolactose is what binds to the repressor and removes it from the operator sequence, okay? So essentially, if the IPTG is absent, okay, our repressor isn't gonna get taken off of the operator sequence. So we have to have the IPTG in order for us to be able to transcribe anything in this situation. Okay, so for part A, it says that IPTG is absent. We automatically know that it's not going to transcribe anything, so we can assume that it's not going to be blue. A little more information though, the X-gal, what that does is essentially if anything is transcribed, the beta-galactosidase will react with the X-gal to form blue E. coli cells. Okay, so with that information, we know that if X-gal is present, the cell can be blue. Okay, so on part B, we have IPTG present, glucose is absent, meaning that we need to metabolize lactose. So we are going to transcribe the sequence, and then X-gal is present, so when that gets transcribed, it'll turn the E. coli cell blue. So B is the cell will be blue. On part C, IPTG is present, glucose is absent, and X-gal is absent. Because the X-gal is absent, we will transcribe the sequence, but the cell won't turn blue. Okay. On part D, the IPTG is present, so it's taking off the repressor. Glucose is present. So because glucose is present, we won't need to transcribe the sequence in order to metabolize lactose. So because we have glucose, we don't need to transcribe. So for that case, we won't transcribe, meaning that it won't turn blue. For part E, IPTG is present, glucose is still present. So same reasoning as D, it's not going to be blue, but G-gal is also absent. So even if we could transcribe, it wouldn't be blue anyway. So essentially for A, not blue, B, blue, C, D, and E are all not blue as well.
Perfect. Okay, so part 4, what we're trying to do is just figure out whether they're inducible, non-inducible, constitutive, and whatnot. And so for part A, we have these parts right here, and so from this we know that there's no lactose, and because there's no lactose, transcription can occur, and so that one's inducible. For part B, we know this is non-inducible, just because we don't know whether the presence of lactose is there or not, and so it cannot be transcribed, and so therefore it's non-inducible. And then for part C, is constitutive. Part D is non-inducible, just kind of for the same reasons. And then part E is constitutive as well.
All right, so that brings us to question 5. And in this one, it's referring to the experiment that we read about in our reading this week. And it's kind of like saying, if these guys had used the genotypes listed below and then put them in 2 different test tubes, one with lactose and one without lactose, our job is to figure out how much yellow product would be present in each tube. So in part A, in the presence of lactose, you're going to have a level 0, so you're not going to have any yellow color at all, as well as in without lactose, you won't have any yellow at all either. For part B, you'll have a grade of 1 for in the presence of lactose, and in the absence of lactose, you'll also have the level 1 yellow. Part C, in the presence of lactose, will be level 2, and that's because you've got it on the chromosome and the plasmid. And then in the absence of lactose, it'll be level 1. For part D, in the presence of lactose, will be 1. In the absence of lactose will be 0. And part E, in the presence of lactose, it will also be 0, level 0, no yellow at all. And in the absence of lactose, you will also have no yellow. It'd be 0.
Thank you.
Bye.
Bye.